all my dear students. This is our topic from first unit, structural organization of bacteria. Before entering this topic, we should know about some basic things. What is mean by microorganism, what is eukaryotic cells, prokaryotic cells, and what is bacteria. First of all, we should know what is mean by microorganism. Microorganism, it is a small organism we can't see in our naked eye. We can view under one microscope. And then eukaryotic cells. This is the main thing. The word itself we can know eukaryotic. Eu in the sense it have the true cells, true nucleus, which is bounded by one membrane. Each and every organs it is bounded by one membrane, that is eukaryotic cells. Example for this is protozoa, fungi, plant and animal. It may be single or multi-cell. That is eukaryotic cell. Next is prokaryotic cells. It is unicellular organism. Example for this is bacteria and archaea. Prokaryotic in the sense it look like oid in the sense it won't have any true nucleus. It have some nucleoids, that is like nucleus like structure, that is nucleoid. Example for this is bacteria. Next, bacteria. In 1676, Anton von Leeuwenhoek, he is the father of microscope. He only seen the first structure of bacteria in 1676. It is the single celled organism, and this is the prokaryotic cells. It won't have any true nucleus, and we can view this in microscope. This is the general structure of bacteria. Here we can see some parts of bacteria. One is flagella, capsule. Next one is cell wall, pili, plasmid DNA, cytoplasmic, ribosome, polyribosomes, chromosomal DNA. These are the basic structure of bacteria. And next one is shapes of bacteria. Normally we can differentiate based on their shape. One is spherical, rod, third one is spiral. Spherical, for example, spherical shaped bacteria, staphylococci, that is the spherical shaped bacteria. Rod shaped bacillus, lactobacillus, that is rod shaped bacteria. Spirillum, like spiral shaped bacteria. And next one is comma shaped bacteria, that is example for that is Vibrio cholerae. Next one is bacterial cell structure. We can classify the bacterial cell structure based on three categories. One is internal structure, which is present inside the cell envelope, which is the over cover of the cell. Next one is external structure, which is present outside the cell wall. Internal cellular structure. Some important internal cellular structures are cytoplasm, chromosomes, nucleoid, plasmid, ribosomes and inclusive bodies. First one is cytoplasm. Cytoplasm, this is present inside the plasma membrane. It look like some gel matrix, gel-like structure. It won't like solid-like structure. It look like gel-like structure. We can view this in electron microscope without any featureless, somewhat water-like structure. We can view in electron microscope. Some constituent present in cytoplasmic are protein, vitamins, ions, nucleic acid, amino acid, sugar, carbohydrate, fatty acid and their derivatives. Next one is nucleoid. Before itself I told nucleoid, oid in the sense it's like structure, not true. Nucleoid is a nucleus-like structure, it's not a true nucleus. And DNA is present in this nucleoid and some cytoplasmic membrane which is covered outer layer of this nucleoid. Next one is plasmid. Plasmid, this is the very small chromosomal DNA. When compared with chromosomal DNA, the size of the plasmid DNA is 1 to 200 kilobyte. Normally chromosomal DNA, it have 4000 kilobase pairs. Next one is plasmid function. Plasmid function, it has some several functions like antibiotic resistance power. It gives some resistance power against antibiotic and conjugate plasmids. Conjugate plasmids, it helps to exchange the DNA from bacterial cell to another bacterial cell. Next one is ribosome. This is the manufacturing area of protein which consists of RNA and protein. This is abundant in cytoplasm. We can see single ribosomes or multi-ribosomes. That multi-ribosomes are called polyribosomes. Next one is bacterial chromosome. As we have already seen, plasmid DNA is a very small chromosomal DNA. Bacterial chromosome, it is a somewhat big when compared with plasmid DNA, and it has some base pairs. What are they are? Adenine, cytosine, thymine and guanine. This is bounded by hydrogen bond. Next one is inclusion bodies. It won't have any specific function, but it will be present in bacteria. And it won't involve in any metabolic activities, but it is necessary to present in bacterial cell. Cell envelope, already we had seen some internal structure of the bacteria. Next one is cell envelope, plasma membrane, periplasmic space, cell wall, outer membrane. These are the cell envelope present in the outer layer of the bacterial cell. Plasma membrane. This is the outer layer of the cell. This have the two phospholipid bilayer, that is double layer phospholipid bilayer. It is consists of lipid and protein. It won't allow any water inside because of it is hydrophobic in nature. So it won't allow any molecules inside the cell outside without any permission. Next one is periplasmic space. The space between cell wall and the cytoplasmic membrane and the cell wall from the lipoplasmic layer. This two space, that is perispasmic space, and next one is in gram positive we can see the space between cell membrane and the periplasmic space. This is the space between the cell membrane and the outer layer of the cell, that is periplasmic space. Next one is cell wall. This gives the perfect shape to the bacteria. As we have already seen, spherical, rod shape and spiral shape. This is the major role to give the specific shape of the bacteria, and it is consist of peptidoglycan. Peptidoglycan is the combination of polysaccharide and protein. Mostly in Mycoplasma bacteria, there is no cell wall present in this particular bacteria. Outer membrane. This outer membrane is present only in gram-negative bacteria. This is the outermost layer of the gram-negative bacteria, which is consist of lipopolysaccharide, lipid plus polysaccharides. This is similar to the plasma membrane, but it is very less permeable. It won't allow any material inside the cell, and it has some porins, thinning holes. It allows only some minute particle, but it won't allow any chemical or antibiotic inside the bacteria. External structure in the sense outermost part of the cell, that is flagella, pili and glycocaly. Flagella, it acts as a propeller in the sense it helps to move from one place to another place. It acts as a propeller on the ship, and it is made up of flagellin protein. In singular we can tell flagellum, but mostly in bacteria it have n number of flagella, so we can call it as flagella. It have 20 nanometer in diameter and 20 micrometer in length. Based on their arrangement, we can classify this into 5 types. One is monotrichous, locotrichous, amphitrichous, peritrichous and atrichous. Monotrichous in the sense, in the single end of the bacteria, it have one flagella. Locotrichous, in single end, it have the cluster of flagella. Amphitrichous, it have cluster of bacteria in both end. Peritrichous, flagella, we can see that flagella in the entire surface of the bacteria. Atrichous in the sense, there is no flagella present in the outermost layer of the bacteria. Next one is pili. Pili is the small hair-like structure. We can view this clearly under electron microscope, we can see some hair-like structure present outermost area of the bacteria, and the diameter of this pili is 2 to 10 nanometer, and length of this pili is several micrometers in length. We can see this in outermost area. And next one is glycocali. Glycocali, it is the sticky coating which we can find out outermost layer of the bacteria. It is made up of polysaccharide, sugar and protein, and we can classify this into two types. One is rigid capsules and another one is slimy area. We can see this in pathogenic bacteria like Vibrio cholerae, Staphylococcus. Like this, we can view this glycocali in several bacteria alone. And this is the possible questions: difference between eukaryotic cells and prokaryotic cells, and gram-positive bacteria and gram-negative bacteria. This is the main important question. We should know the difference between gram-positive and gram-negative. Using what technique? Staining technique. We should know that staining technique and bacterial cell structure, which we had seen previously about internal structure, external and inner layer of the cell. Thank you.